it's a really, really great pleasure to introduce our next speaker, which is Dr. Mircea Podar. Mircea received his PhD from the University of Texas Southwestern Medical Center, and then went to Woods Hole for his postdoc, then to the Salk Institute, before he joined Diversa. And actually, I think it was when he was at Diversa, which was almost 20 years ago when we first met, which is hard to think about. He joined Oak Ridge National Laboratory in 2007, where he's a distinguished staff scientist in the biosciences division. When I think about Mircea's research, what strikes me always that it's extremely multifaceted. It covers human health, the environment, and specifically he's applying very diverse and very cutting edge technologies, experimental technologies and approaches. So without further ado, let's hear from Mircea.
Thank you, Tanya. Thank you, Nigel, and the organizers for inviting me. And I'm always thrilled to come to JGI. As Tanya said, I think over the last 20 plus years, we've been collaborating, interacting. I have so many friends here. In a way, we kind of grew up, our science grew up together. Of course, mine on a very different, smaller scale. But anyway, it's always a pleasure to be here. And I thought this is very appropriate to start with this slide, which shows the sort of the wild side of the microbes. And we were talking last night actually with Bruce that we both use the word wild in our presentations, but actually it's the first time I've used the word wild, and it's coincidental. I usually say dark microbial matter, things like this. But so 20 years ago, there's a phylogenetic tree on the left from Phil Hugenholtz when he was in Norm Space Lab here at Berkeley. And that shows the diversity known at that time of the bacteria. And it was known purely on the basis of ribosomal RNA sequences. And if you see in there those white widgets, those are all lineages that nobody knew how they were. They were, again, discovered based on ribosomal RNA sequences. They received some funny names like TM6, TM7, OP5, and so on. And if we move 2 decades further, the tree from Jill Banfield's lab, also here at Berkeley, which dramatically expanded, it shows the expansion of the microbial diversity. And this tree is now primarily based on metagenomes and single cell genomes. So they're not only based on ribosomal data. And now if you look at all those little red dots, those are microbial lineages that are only known based on genomic sequence. And for most of them, they remained in that status of uncultured. Some people call them unculturable, which a term that I really don't like to use. And regardless of their names and how this tree has been evolving since 2018, it still remains true that there's a vast majority of microbes out there that we only know based on their DNA sequence. And to go back a little bit in time, what's interesting and what actually kind of moved me from my original training as a molecular biologist and biochemist into the microbiology was a discovery also 20 plus years ago from Karl Stetter's lab in Germany. We actually discovered a new lineage of microbes, of archaea, based on cultivation. So that's probably the last major lineage of life that actually was found based on cultures, not based on sequence. And what was remarkable about it was that that particular organism, is this on? Do I need a microphone? I don't even know. Is it on? Oh, okay. So those little red dots that you see, those were those microbes, a nanoarchaeum equitans, a hyperthermophilic archaeon. And they strictly dependent on association with another archaeon, Ichnicoccus. And that type of association was really unique at that time. And I've been fascinated by those organisms for many years, and we've studied them in my lab, and we've discovered some new ones in Yellowstone, for example. But there's some interesting, as Bruce was saying, microbes don't live on their own. They interact with others. And in this case, this little nanoarchaeum, it's an ectobiont. One could call it a parasite. And as you see in that scanning EM that one of my postdocs did a few years ago, so the little ones, once they deplete the resources of their host, they are actually reaching out. They can actually detach and somehow find new hosts and attach to them. So in a way, you could imagine them as like viruses. Of course, they're not viruses, but you could make an analogy. And what we've found in the lab was that they could actually even potentially jump the species barrier and infect related hosts. So all those interesting mechanisms, we studied them using a variety of omics. And what I can tell you that we still actually have no clue how they do it. You see those appendages. We don't know what proteins they're involved. We don't know how they're sensing their host. What's the specificity? We basically can look at them and wonder, but we need better approaches to actually dissect their biology. And there's, as Bruce also mentioned, there's a lot of ways microbes interact in their communities. And probably a lot of those drive our inability to culture and isolate them. So they can compete with each other, they can help each other, they can cross-feed. Some of them require interaction with other microbes. So they're like that nanoarchaeum. Some of them grow very slow, and maybe that's why we can't really grow them. Of course, some of them can be dependent on eukaryotic hosts like plants or animals. Many of them may even have novel type of metabolism that we really cannot predict. And they have different cellular structures. So there's a lot of factors out there that are actually hidden to us, even though we can sequence them. So over the year, of course, I've been involved in all the aspects around characterizing microbes in communities. And of course, we all do now like to go out there and take the samples and do either single cell genomics or metagenomics, assemble genomes, SAGs and MAGs, and then predict their metabolism, and then somehow try to transpose that back to their function in their natural communities. But really, when we were doing this, and I was doing a lot of single cell sequencing, and a lot of that is, for many microbes, especially the very rare ones, it's kind of a lottery. You'd never know what you're gonna get. But the question really is there a way to get back from that genome and that model prediction and really have that microbe in the lab and really start to study that in isolation if you want to look at ultrastructure, things like that, but you could also assemble them, put them together, as Bruce was also showing, the reconstituted communities. So in theory, we have all that we need, right? We have the genome, we have the blueprint. Can we use that blueprint to actually isolate that microbe? In theory, it should be doable. In practice, it's not that easy. So what I was going to show you today, of course, I hope I convince you the why we need to do that, but also maybe to start getting into the how we could potentially do that. And it's not only for uncultured microbes. Even if one is interested in a microbe in specific, in a very diverse community, for the vast majority of them, we don't have specific ways to isolate microbes. Maybe for phototrophs, maybe for chemolithoautotrophs, you can design methods to kind of weed out the others. But in general, if you're interested in a particular microbe and there's 10,000 other ones out there, there's really not a good way to get to it. So that's kind of like one of the drivers of thinking of maybe can we get to that in a way, the same way you could get by PCR to fish out a gene from a metagenome without having to sequence the whole thing. Can we come up with something that will allow us to do this at a microbiology level for microbes, not only for genes? So there have to be, of course, a number of criteria that have to be satisfied. One of them, we have to find our way to achieve that specificity. And that specificity can be directed to a specific strain or a group of microbes. So can we tune that the same way as you do PCR and you can design very specific primers or degenerate primers, things like that. we also need a way to be very sensitive. So if your microbe is 0.001% of that community, how can you get to that? Because if you do it randomly, you're never going to get to that particular microbe. So can we come up with a way to achieve sensitivity? Now, if we actually want to grow that microbe, not only to sequence it, I mean, of course, we could also do this for sequencing only. But if we want to grow it, everything we need to do to that microbe or sample needs to retain that viability. So can we achieve the two components without killing that microbe, without fixing it? And then, of course, ideally, can we scale this up? So in the past, when you had a single PI lab and you wanted to sequence a genome, you had to make all those libraries, put them on Sanger. There was a whole PhD project just to get to a genome. Nowadays, you can scale this up, multiplex this, and you can get 1,000 genomes in a few days, right? Can we achieve that at high throughput at microbial level? And one solution that some years ago I was like thinking like, well, maybe we can do that. And it's not really something dramatically novel. It's the use of the molecules that people in biomedical area and research use all the time. And those are antibodies. And those could be natural antibodies raised in animals, or nowadays you can develop actually synthetic libraries. And you can use those as hooks. And then if you can combine those with a high throughput approach like flow cytometry, potentially you can meet all of those criteria that I just had up there. So we came up with this concept of, we call targeted reverse genomics, in which how can you achieve especially the retaining viability aspect, because you don't want to get inside the cell. Once you get inside the cell, you essentially kill the cell because you fixed it. So one way to do that is by focusing on the surface of the cell. And if we want to achieve that specificity and sensitivity, all we would need to do is identify surface markers. Proteins would be the best. And then design antibodies or nanobodies, for example. Nowadays, we switch to these synthetic nanobodies that will target predicted surface proteins. And if you pick the right proteins, you can achieve the selectivity and you can achieve the sensitivity. Of course, there's some ifs there, but if we are good at analyzing genomes and metagenomes, in theory, we should come up with ways to do that and scale it up. And then once you have such hooks, they can be labeled, they can be applied to any sample you can think of. Either sort individual single cells, you could sequence them, confirm that you get what you want, and then come up with, move to the next hard part. Like if you have now the cells, can you put them in a condition where they will grow? And for the microbes that require interaction with other microbes, they will also bring along their hosts. So if you find the right conditions to grow their host, you then can find potentially the right conditions to grow anything. So, and then characterize them and so on. So that's kind of my dream for the next phase of microbiology, is that one could apply this in a way that there is no such thing as unculturable microbe. So to develop this whole thing, we've actually used the human oral microbiome. This concept we raised the interest of NIH in applying and developing this. So we used it on trying to isolate some dark microbial matter from the mouth. And we all share some of these lineages. For example, TM7, which are now called Sakari bacteria. And then there's other ones like GNO2 and SR1, and they all have fancier names, but they're all very low abundance. So for the SR1 and GNO2, you can have like 0.01% of your microbial community in the mouth are those microbes. So they all have reduced genomes. Potentially, they all depend on interaction with other species. So that's how what we focused on. And to make a long story short, for TM7, for example, we've selected one of those potential proteins that was predicted to be on the surface based on a single cell genome sequence that was sequenced a long time ago in the Relman lab. And we isolated and achieved cultivation of that particular organism with one of its hosts, which is a standard actinobacterium in the human mouth. And we and others have done a lot of work on this, and there's fascinating biology. They are potentially also involved in health and disease. Not going to have time to get into that. But what they also show is this very interesting host specificity, like I was telling you about the nanoarchaeum. So on the left, there's a phylogenetic tree of TM7, oral TM7. On the right, it's a phylogenetic tree of actinobacteria that they can use as a host. And you can see that the different TM7 can use potentially different actinobacteria as a host. What is their specificity? What is the mechanism? We have no idea how they recognize and interact with each other. This also actually occurs out there in nature, and there's a paper published by the Batinovic et al. From a lab in Australia, where they've isolated one of these TM7 using traditional microbiology, but they were looking for viruses. They were looking for plaque-forming viruses, and they got a plaque-forming bacterium, and that particular TM7 Mycosimbacter can infect a wide range of actinobacteria, and it's potentially useful in controlling these biofilms in water treatment plants. So we have now bacteria that actually almost work like lytic phages. So this can have a huge influence on the microbial communities. And TM7, for example, occurs pretty much everywhere, in soil, in water, in the ocean, in the human mouth, you name it. We've also worked on developing such approaches, this approach for the other microbes, SR1, GNO2 using synthetic nanobodies. And we have some ideas on who might be their potential hosts. Like Bruce was also mentioning these, another Vampiro Vibrio, but it's similar. They basically can modulate the dynamics of their host communities in their natural environment. And one of these has been cultured by a lab in Russia, and it's called Vampirococcus, and also attacks and lyses its hosts. So we are essentially using the same reverse genomic approach to try to get to not only the oral ones, but other ones. And there's some models based on microscopy and genomic data that suggest that some of these interactions are mediated either by type 4 secretion pili or by these giant S-layer proteins that some of these microbes have. So in collaboration with our proteomics colleagues at Oak Ridge, we actually verified that, for example, T4 pili for TM7 are very highly expressed proteins, and we have developed nanobodies against them, and we are using those to either fish out additional TM7 or to use them in a genetic screen to identify the genes involved in that particular interaction. And on the side of SR1, for example, we have another collaboration funded by an NSF grant with Noha Youssef and Mustafa El Shahed at Oklahoma State, where we're looking at a sulfidic spring in Oklahoma where there's a lot of these lineages of uncultured microbes. So we're focusing on this SR1 in the Zodelton spring. And in particular on one of those S-layer proteins, which also we identified to be very highly expressed. It's actually the most abundant protein in those microbes. So we've also generated nanobodies for those types of, for those proteins. And we're working on trying to use them to isolate those particular SR1 and their hosts. Now, if one, so far I've been telling you about microbes that we're interested in because of their taxonomy, because of their evolutionary place in the tree of life. But as Bruce also mentioned, for some, in some cases, what we want is, we don't really care who are the microbes. We care which microbes respond to different types of environmental stimuli. And we're not using SIP in this case, but as part of a project we have at Oak Ridge on studying the interaction between microbes and populus trees, we also want to understand how microbes respond to chemical stimuli that the populus root, for example, produces. So every plant produces these exometabolites, and then the microbes can use them as food, for example. Now, the plant potentially uses those to attract specific types of microbes that would be beneficial for the plant, right? So if you want to attract your friends, try to be specific and attract specific types of friends. So in order to identify who are potential players in this interaction between plant and microbes, we're actually using a different approach called BONCAT. I know that some of you are using that, in which you're essentially providing the microbes with different types of food, different types of metabolites, and then you look to see who responds to that, and who responds to that based on protein synthesis that those microbes activate. So those microbes would incorporate a certain type of amino acid derivative, and that can be then labeled using click chemistry. So a lot of people do this, but one of the things we thought is like, well, it would be best if we can not only identify who they are, can we also, again, get them and grow them? So in order to do that, of course, you need to make it without killing the cells. So we are using this BONCAT combined with surface labeling with biotin, and then potentially we can get those microbes either using flow cytometry or magnetic beads, and not only sequence and see who they are, but actually grow them in culture and further study them. So I'm just showing here a proof of concept that that particular approach actually works. We've grown in the lab individual strains of these microbes under those particular conditions, labeled them with the clickable biotin, and then added them into an actual natural community sample. On the right, we looked at how that changed the composition of that community. And then in this particular example, just use a simple magnetic enrichment, and we were able to retrieve a lot of those microbes that we added in. So essentially, they get those proteins on the surface labeled, and we're able to retrieve them. So we want to use this approach and concept to identify microbes that would respond specifically to certain type of plant-derived metabolites. So I try to give a really fast overview of this concept of using the genomic information to get to microbial lineages of interest, whether that's based on phylogenetic position in the tree of life, or whether that is based on potentially physiological responses to environmental stimuli. And especially for the microbes that specifically and obligatively, in some cases, interact with other microbes, this really, I think, opens opportunities to understand mechanisms, to start to dissect how these microbes talk to each other, how they evolved together, what kind of structures and chemical stimuli evolve. And a lot of these, I think, you know, using just pure cultivation independent, you can get some clues, but you cannot really prove mechanisms unless you are able to grow them and look at them. And because of these, all these techniques at the right place can be scaled up in throughput, right? Gene synthesis can be done in high throughput, not only one at a time. Protein expression can be done in high throughput. Nanobody identification, selection, characterization can be done in high throughput. So in theory, this could be done in a way that you could like go after dozens and dozens of taxa at the same time, rather than just doing it one at a time. And then, of course, a lot of these microbes that we would end up having in our hand can have a wide range of applications, biotechnology, biomedical, environmental, and so on. And I will end up here. There's a lot of people, of course, that I have to thank, and I'm grateful to. At Oak Ridge, many of the people that were involved in the human microbiome aspect and the original development have since left to other places. Ohio State collaborators on the biomedical field. Of course, like I said, I've interacted with JGI and the friends here for many years on single cell, on data analysis, on metagenomes, and so on. And then other collaborators that I have at Oklahoma on Zoduton Spring, and of course, the entire PMI project team at ORNL involved in studying the interaction between microbes and the plants. And then, of course, a big thank to the people that paid the bill on these, starting with NIH, NSF, and last but not least, DOE, who has been funding our activities as part of the PMI project, but other projects as well. So I'll be happy to take questions if there is time. Thank you.